This is the Discovery Files podcast from the U.S. National Science Foundation. As the wheel of the year turns, we transition into cooler weather and falling leaves, entering the season where kids are returning to school and the adults to the office. A pair of scenarios that allow the cold and flu season to flourish. To learn some strategies for protecting ourselves and learn more about viruses, we spoke with microbiologist Joanna Shisler, NSF Program Director in the Directorate for Biological Sciences, Division of Integrative and Organismal Systems, with a primary interest in virology. Dr. Shisler, let's start at the basics. What is a virus?
So I like to define viruses as very small biological particles that are consisting of genetic material, so genes, that are protected by a protein-based coat. There's an amazing number of different types of viruses. So the genetic material that some viruses have is DNA. So that's something that you and I have that makes up our own genomes. Other viruses have RNA, and that's a little bit different. And then there are also viruses that either just have that nice shiny capsid, whereas other viruses will take parts of its host cell membrane and have it wrap around that nice capsid. And so what you see is a really amazing variety of viruses when it comes to shapes and sizes, and that is some of what virologists originally used as a way for a basis for naming viruses and for categorizing viruses.
Are viruses rare?
So we used to think of viruses as very rare things that would happen to us, right? So normally we don't have any viruses except for when we come down with a cold. But what we're starting to understand is that actually there are a lot of viruses that are in different parts of our body and at different times. And so there's estimates that there are around 380 trillion viruses either inside or on the surface of our bodies at any time. And so it's really fascinating because we're thinking about trillions of viruses, and when you think about our own microbiomes, you're thinking about billions of bacterial cells. So when you think about yourself as a whole, there's actually probably more viruses and bacteria than there are human cells that make you up.
Do we know where viruses come from?
No, we do not know where viruses come from. The field is working on that, and there are a couple of different hypotheses that people like to think about. One is that perhaps the genetic material and proteins kind of got together to form viruses right around the times when the first cells evolved on Earth. Another idea is that viruses evolved from pieces of DNA that are called plasmids. And these are tiny little circular DNAs that sit outside of the host genome.
Are there good viruses?
So of course there are good viruses, and I'll tell you a couple of examples of how researchers have taken viruses and turned them into something good. So there are a group of viruses called oncolytic viruses. Onco, think about cancer, and lytic meaning to lyse or to kill. And so people have been trying to take normal human viruses and change them just a little bit so that they specifically target and kill cancer cells. There are other people who have used bacteriophage, which are a type of virus that kills bacteria. So we've been hearing a lot about antimicrobial resistance and how there are bacteria that can become resistant to antibiotics. And so phage are one way people are thinking about being able to lyse bacteria. Then in addition, we're starting to get an appreciation for how viruses can actually be helpful to us as humans. So there's a really neat example of an ancient retrovirus. We have an ancient retrovirus in all of us that produces a syncytial protein, and this protein is important for development of the placenta. And then we're still just now figuring out how viruses of the microbiome can actually be helpful in things like helping us process food and nutrition.
What do you do when you see a report about a viral outbreak?
I like to try to find the original article that they're talking about. And so sometimes you can find that article by doing a search. The other thing I do is that I look to other health agencies to find more information. And so you can look at global health agencies, such as the World Health Organization, You can look at some of our own federal health agencies, such as the Centers for Disease Control, or you can look to your local public health districts or even to medical schools, for example, the Mayo Clinic.
How can we avoid catching viruses and getting sick?
There's only a few ways that viruses can spread. With COVID, we've learned about respiratory droplets and how important that is. So if you're thinking about how to protect yourself from respiratory viruses, then you want to think about covering your cough or wearing a mask or sneezing into a tissue and then throwing that tissue away. Viruses can also be spread by the fecal-oral route. So that would mean viruses that replicate in the intestine and come out in feces, and then people are not very clean with their hands, and they'll then put their hands to their mouths or their eyeballs. So the best way to protect against viruses that spread through that route are by washing your hands or not touching your face.
How can we avoid the next pandemic?
So the way that we've been detecting these viruses is through sequencing of genomes. And the ability to sequence genomes has become much more reasonable, and we've been able to do it from many more locations. And we've also been able to have technology that allows us to detect very few numbers of viruses that are in a sample. And so I think where we are here in society is we're at this amazing ability to be able to sample sewage water and quickly sequence the material in that sewage and detect if there's viruses or not. People are now working on being able to collect viruses from the air. They talk about the air virome to be able to detect what's in the air. So I think we're at an amazing time where we can sample and also detect viruses at a very early stage, even before humans or other animals become sick.
We also spoke with Professor Gerri Botte, Whitaker Department Chair in the Department of Chemical Engineering at Texas Tech University, who developed a COVID-19 sensor that detects spike proteins at the early stage of infection during the height of the pandemic. Dr. Botte, how did you get started on the ultra-fast COVID-19 sensor?
Basically, when the COVID-19 pandemic started, I was working on sensors related to E. coli. And then I spoke with my graduate student. We were all like low-key in drying houses and say, hey, is there any way we could do something with our E. coli sensor platform chemistry that we know and how we can see if it works with COVID?
How does it work?
So the way this works is that we found something very unique. So when we apply basically an electric field into the surface of a catalyst, what we observe is that the spike protein has a very unique affinity to this catalyst. And in fact, the response of that catalyst is supposed to be completely flat, not to see the spike. And this spike was kind of like unexpected. It also takes place in a very, very short time. I'm talking about milliseconds, okay? And so what we were able to demonstrate is that there is an interaction in between the spike protein that is present in the SARS-CoV-2 and these electrocatalysts when we apply an electric field in a specific electrolyte. So, and then we took that basic science and put it into a device. And then further than that, we were able to demonstrate that we could do it with saliva samples. And then we had taken that device in order to collect from air. And we were able to respond very fast in order to contribute to the solution of a major challenge such as the COVID-19 pandemic.
And I understand there's already been a next step with the device.
So today, these devices has been become into some sort of now what it calls more like a manufacturing system. And it's being expanded as a platform for other viruses, not only to detection for saliva, but also to detect from air. And we're integrating not only electrocatalysis and electrochemistry, but also deep learning and machine learning algorithms in order to do this categorization.
Special thanks to Joanna Shisler, Gerri Botte, Gino Scofidio, Adam Eggers, and Dina Headley. For the Discovery Files, I'm Nate Potker. Please subscribe wherever you get podcasts, and if you like our program, share with a friend, and consider leaving a review. Discover how the U.S. National Science Foundation is advancing research at nsf.gov.